Hi, this is Mr. McLean, and what I want to take a look at right now is some of the details of how DNA replication processes take place. So we're going to focus in on a lot of the details of Chapter 16. One of the first things I wanted to point out is we understand, based on some experimental evidence, that the mechanism for DNA replication is the semi-conservative replication model. And what that means is that whenever we take the original DNA molecule, which is shown with the dark blue, after the first replication, what we're going to find is that you're going to have half old and half new. And you're going to get the same thing in both of these strands. And then when we go to the next replication, which in this case would be replication number 2, you're going to notice that this dark blue strand will appear up here, and with it will be a new complementary light blue strand. And the same thing is going to happen here. One of these light blue strands is going to be shown here, and there's going to be a new complementary strand with that. And you're going to get the exact same thing here. Okay? And how we came to that conclusion, if we take a look at this particular experiment here, what we're going to notice is that when we look at the way in which the replication took place with the heavy isotope 15 being the dark blue and the lighter isotope of nitrogen 14 being the light blue, and using a centrifuge to be able to separate that and check by density, what we found is that when we look at the first replication, we found a single band. And that single band showed that there was equal density in both of those molecules, which would indicate that half of them were old and half of them were new. So it wasn't as dense as if it were all nitrogen 15 or as light as if it were 14. So it's kind of a hybrid. And then when we did our second replication, what we found is there were actually going to be 2 bands. So the one up here is going to represent a lighter band, and the reason why that's going to be less dense is in this second round of replication, all you're going to find is nitrogen 14 isotope, whereas this other band represents the other pair of DNA molecules where they're still going to show some of the original nitrogen 15 coming through along with the new nitrogen 14. So the idea is that our replication is semi-conservative, and the idea is that we're going to be able to see old and new coming together. So to kind of highlight a few of the details of how replication takes place, we want to focus in on this particular image. If we look at it, what we do is we take what's referred to as an origin of replication. And the origin of replication creates a replication bubble. Within our bubble, what we end up with are 2 replication forks. So we would actually have a replication fork here, replication fork there, and the idea is that along the length of a DNA molecule, you're going to find lots of origins of replication. So the speed of replication is definitely faster because it's happening at multiple points. So if we zoom in on one of these areas, the other thing that we want to take a look at is it's going to create this type of arrangement. Okay, and we're going to highlight the pace of replication and focus in on some of the key enzymes. So if we take a look at what a replication fork is designed as, it says it's a Y-shaped region where the DNA strands are actually elongating, and we're going to see that the primary enzyme for opening things up is DNA helicase, which is going to go through and it's going to unzip by breaking hydrogen bonds. That's going to open up that DNA molecule. And we're also going to notice that there are proteins that are going to be used to keep that DNA molecule from tightening back up and closing back up. Those are the single-stranded binding proteins. And then the other enzyme that's critical is topoisomerase, which is going to kind of work ahead of that replication fork. And what it's going to do is it's going to break some of those bonds that allow the DNA to kind of relax and swivel and then rejoin, as opposed to becoming overly tightened. So in this particular scenario, we can see the direction that this is moving is kind of towards the left side of our screen. Here's our helicase, and the helicase really looks like it's working along the lines of a zipper. As the helicase is moving, these bonds between these nitrogen bases are being separated. Topoisomerase, you can kind of visualize right here. It's making these little cuts and breaks in that molecule to prevent the over-tightening. And like I said, the topoisomerase just works a little bit ahead of where the helicase is at. After we've opened these areas, these gray molecules right here are the single-stranded binding proteins. The idea is, because of the way in which they were bonded initially, when we break those hydrogen bonds, they're going to want to reestablish themselves. So by putting the single-stranded binding proteins in there, what that's going to do is that's going to keep it from basically closing back up. Now, when we actually start to create the DNA molecule, one of the things that we need to keep in mind is that the DNA replication is always going to operate in such a way that we create a new strand in a growing 3' direction. Okay, so when we think about our DNA molecule, we know that the orientation of our DNA molecule is such that it's anti-parallel. Okay, and we have some images here. Let's take a look right here. So if we look at a DNA molecule and we recognize that they are oriented in opposite directions. So we have on one strand, you're going to have what's referred to as the 5' end. We name it the 5' end is because the attachment point, 1, 2, 3, 4, and here's my 5th carbon right there. And that's where I attach my phosphate onto. So the attachment point at that particular position when you're facing that way is actually in the 5th position. If you take a look down here, we call this 3'. And the reason why that's in the 3rd position is position 1, position 2, position 3. Okay, so the way in which this is going to allow for additional nucleotides to come into the system is that we are going to be able to bring in what's referred to as a nucleoside triphosphate, which is going to have 3 phosphates on here. It's going to have our 5-carbon sugar. It's going to have hydroxyl group there. It's going to have a nitrogen base. And whenever they are brought in, the enzyme that's going to catalyze this reaction is DNA polymerase. DNA polymerase is going to catalyze a reaction that will release what's referred to as a pyrophosphate, which means that 2 of these phosphates are going to come out, and we're going to be able to make this chemical reaction take place. So now what we've done is we've added a new nucleotide. Now this only works in a growing 3' direction. So as we take a look at these 2 strands, this is serving as our template right here. So the template is oriented with a 3 up here and a 5 there. Our direction of movement, if we're moving from the top of the screen to the bottom, is allowing us to continuously add, because this end on the new strand is 3 and 5 would be towards the top of our screen. So if we go back and we look at this picture right here, what we notice is that on this top strand, we see a 3' orientation. So whatever letters I were to put in there. So if I said this was A and this was T and this was C and this was G and this was A and C and C and A. Whenever we start to add the complementary nucleotides eventually, what we're going to notice is that it will add and build this new strand in an anti-parallel arrangement to that. So if this strand was 3 at that end, the new strand is going to be 5, and that means the 3 would be down here. So if you have 3 in the direction where the overall replication is occurring, which is towards the left on our screen because we're creating this fork at this particular point, then we're going to be able to go in a continuous fashion. So our first step, you might notice, is going to involve this red structure, which we have listed as an RNA primer. So let me clean out a little bit of this writing that I have here so we can kind of focus in on the original picture. So we have this RNA primer. An RNA primer is actually a very small sequence of RNA nucleotides. And in order for us to go through there and put down DNA, we actually have to establish a small RNA primer first. Enzyme that's responsible for that is primase. So the primer gets laid down, and then from that point on, we're going to use DNA polymerase 3. Okay, so let me jump ahead. I know I have a slide right up here that shows us this. Okay, so here's our primer. After we lay down our primer, here comes my DNA polymerase 3, which is going to be represented by this enzyme. And you can see that it's growing in the 3' direction. Now we also have something called the sliding clamp that's going behind it, and it's literally sliding and clamping these two things together, making sure that we get the bonding that's necessary. So if we look at the bottom image here, you can see that this is, once it's set that primer, it's kind of moving in a continuous direction from 5 moving towards 3. That's our leading strand. A lot of times we look at images like this, and we'll see that the leading strand, which would be this one right here, this is growing towards a 3' direction. So from this perspective, this is 5. There's my primer. Here's my newly established DNA, and it's growing in that fashion. Now, on the other side, let's split this in half. On this portion, we refer to that as the lagging strand. Reason why the lagging strand is a little bit different is the orientation of that particular molecule. The overall direction of what we're trying to do, if we look at this, is right here. Let's zoom in. If we split this screen right here, it's not working for me. If we split this screen right here, and we say that the direction of replication that we're focused on is this side on the left side. Okay, so we already saw that this is nice and continuous after you get that initial primer here. We're trying to grow this way, but the problem is this strand is oriented in the way that it's being established. The new DNA is 3 up here and 5 there. We are not able to add new nucleotides that way, so what we have to do is we have to set down a primer and literally work in an opposite direction. What that does is that creates little fragments. Those fragments make up what we call the Okazaki fragments, and that is the layout for the lagging strand. So let's click over here on this picture right here. So if you're examining this, you'll see that you're going to have you're going to lay down your primer, and then you're going to work backwards from the primer with the DNA polymerase 3. Then it's basically going to work back until it hits the next previous primer. At this point, the replicating machinery is going to be released, and it's going to jump sort of forward if you think about it. So it jumps forward and works backwards, jumps forward and works backwards. What it then does is it's critical that we go through there and remove this RNA. Okay, so what we're going to find is this enzyme right here is going to be DNA polymerase 1. And what that's going to do is it's going to take this RNA primer out of there, and it's going to remove it. And when it removes it, it's going to replace it with DNA. And then you might notice that this has a little gap that remains right here. This gap needs to be sealed up. So this blue thing is my DNA ligase. Okay, that's the DNA glue that kind of holds everything together as we go through this. So big things to keep in mind. If we look at this particular picture, it kind of shows us everything, and this is a good slide to look at. You can see that we have a continuous growing strand on what's referred to as the leading side, and it's growing towards the 3' end away from its 5. And remember, the strand that it's following is exactly opposite, so you can see how it's 5 on the top strand. Down here, because of the orientation, we want to replicate moving this way, but we're actually adding the DNA in fragments coming back this way. Those fragments make up the Okazaki fragments. That's laid down by DNA polymerase 3. We use DNA polymerase 1 to remove the primers, and then we glue the primers together with ligase. So hopefully that helps explain some of the protocols as to how DNA replication takes place.